Welcome. If you are new to Nature's Notebook, I will just give you a quick overview about that. Nature's Notebook is a citizen and professional science program designed and hosted by the USA National Phenology Network. And the National Phenology Network is made up of staff members like Ellen and myself, and several other people who are scientists, researchers, outreach coordinators that are busy curating all of the wonderful data that people submit to our Nature's Notebook citizen science program. And that's an important note that I like to make. A lot of times people think that we are the researchers that are doing climate change research on the data that we receive, but that is not the case. We are here to curate the wonderful data and help to make sure that the data are available freely for other people who are interested in studying anybody's species response to climate change. So that's our role. And let me know if you can't see. Oh, that's going backwards. Let me know if you can't see the slides. You guys should all be able to see the slides. So the reason why we decided to host this webinar is because we do get a lot of questions about our Phenophase definitions and our protocols. And surprisingly enough, they are usually the same types of questions. So the ones that you all submitted here are not different from the ones that we often receive. And that tells us that maybe we need to put this information in a place on the website or provide it to you in different formats so that people get the message pretty immediately about what the protocols are designed to service. So people are also busy trying to interpret what the protocols mean, and that's really not the intent. So we'll describe a little bit about that today. We'll describe why the protocols are the way they are, and we'll describe the way that the data are being collected. So again, the categories usually fall into certain bins about the different parts of the phases, or we also get a lot of questions about the intensity. So we'll try to cover all of those things for you today. And in case you're not aware, we do have all of the information, every single question that people have asked us in the past, including for this webinar and just in emails, shows up in one of our products on the website somewhere. And so that's why people probably feel like they're overwhelmed and they want to have somebody answer their question right away instead of looking through the many pages of resources we have. But I do want to point out that everything is documented somewhere on our website, whether it's in a publication or on the help pages or in the USA-NPN YouTube channel. So the answers to your questions are there, and we also have lots of information about how to use Nature's Notebook online and on the computer. So that's just, I'll give you a little bit of an overview of that at the end. If we have time, I'll give you a tour of the website. So that also means that we are going to be revising how we present this information to you. We're in the process of revising our How to Observe handbook, so some of this information will appear in there. And hopefully in 2020, we'll have some of this more readily available for folks so that they don't have to feel like they're confused when they get started. Okay, so Ellen, I'm going to turn it over to you now, if that's okay, to describe the shape of the data and the database, what we're doing here.
Yeah, and I'll just start by telling you all that I'm the monitoring design and data coordinator for the USA-NPN, and I started probably 11 years ago, just sort of coordinating the development of our protocols, and have been doing it ever since. And I am indeed the one that if you can have questions or don't like it, something's wrong, those come to me. Lorianne, you might be telling Lorianne, but they all do come to me. And so we have seen repeating confusion on certain issues over and over again. And so we're going to try to kind of outline that in this webinar, the common points of confusion. So we'll start with the big picture, the value of the data. The impetus for the Nature's Notebook data collection effort was the desire for a national scale data about the timing of plant and animal life cycle stages that are used in scientific inquiry and decision making surrounding a societal need to understand the environmental changes that we've been seeing in recent decades. And this is, I think, a point that Lorianne's been trying to hammer home for years now. Remember that the protocols are to be taken at face value, and they're designed to be reported consistently. So the goal of this data collection effort is to give the end user of the data a valuable picture of what's happening on the ground with the plant or animal species throughout the season, with all of the phases: leaves, flowers, fruits, eating, feeding, mating, reproduction, rather than to spend a lot of time overthinking whether one leaf on the plant is a young leaf versus an old leaf. That's, you know, we don't need that detailed scale. You can let those minor details go. Just, we want a general picture of what's happening on your plant or with your animal species at your site. So the protocols are designed to capture the basic life stages that the plant and animal species go through during seasons in the United States. They're based primarily on the presence and absence of structures in plants like leaves, flowers, and fruits, and behaviors in animals. So that's breeding, migration, use of different food resources throughout a season or throughout multiple seasons, so that we can document the patterns of these things coming and going in different locations across the United States. And the basic point is for the data to be used. We don't want you just collecting data and it goes nowhere and it's just a wasted effort on all of our parts. But the key thing here is that researchers were only going to use data if they trust the quality and consistency of that data. And since the roots of our program are about scientific inquiry, we work really hard to attain a quality that's acceptable to scientists. And it's very hard to get consistent data from thousands of different observers distributed across a huge nation, especially when we don't pay you guys and we don't send you to a central hands-on training program. So we do the best that we can to try to get consistent data from all you different observers that are out there across the US by providing a standard protocol that we ask everybody to follow. We provide self-serve training resources, as Lori-Ann mentioned. They're scattered all over the website. We invest in engaging local phenology leaders to provide on-the-ground training that resonates with volunteers in their local area. And I imagine that many of you on this call are those local phenology leaders. And all this we can tout as we're conforming to some minimum standard to keep data consistent across the US. We certainly do not have control of what, you know, any given observer is, how they're interpreting it, and we have no way to quality control that really. We just put these things up front as quality assurance measures to try to keep the data coming in as consistent across the board as possible. Okay, all right, so with that in mind, are we on what not to fret? Yep, I'm trying to keep up. I'm looking at a different version that Lori-Ann is, so I'm trying to keep up here. Okay, so first of all, we want everybody to realize that even though we're trying to get science quality data, we have tried to target the phrasing of the protocols, terminology for non-scientists who are willing to learn a little bit more about plant and animal life cycles than an average adult already knows. So if you're one of the many people who do have a scientific background, please don't overthink the phrasing or the intent of the Phenophase definition. Just take them at face value and try to evaluate the Phenophases as you expect a volunteer observer without those years of scientific training or familiarity with botanical or zoological jargon would. So when we say flowers or flower buds, we're not, there's botanical terminology for the word bud, and you know, we do get questions digging deep into that, but we really just mean flower bud in the sense that, you know, somebody understands what a rosebud is. Oh wait, we skipped what to fret. Sorry, I'm on the wrong. Oh, sorry guys. Okay, that was a not to fret one.
You're doing fine.
Okay. All right. Things that we do want to fret. Okay, so follow the official definitions, official Phenophase definitions. Don't change the wording for your local volunteers. This is, like I said, one of the bare minimum ways we try to get consistency across the nation. So we wrote those definitions, and we're very supportive of people making their own kind of supplemental training materials for your volunteers on the ground, but please do not change those official definitions. It's okay to provide additional tips for what your Phenophase might look like for the plants at your site, because there is variation across species ranges, regional levels, and local levels. So it's okay to provide some additional information as to what a flower looks like, but please keep those official definitions intact. Okay, another thing that's important to capture are the transitions when a Phenophase starts and ends. So that's when your reporting goes from a no to a yes. And ideally, we would like everybody to try to catch that no before you catch the first yes happening. And so that means getting out there a little earlier than you think the Phenophase is going to start. And probably also going out a little bit more frequently. If you know that your spring is coming, it's warming, you're about to get the breaking leaf buds, go out a bit early, and instead of going once a week, go every 3 days. That would be great to help get this. And the reason for that is when researchers use the data, they really want to know what the exact date that the Phenophase started was, but we can't give them that because we're not asking people to go out every single day and check every single day. And so what they're looking at is the days between the first yes that you reported and the last no that you reported, and how many days were in between. And so a lot of times we have researchers filter the data. They exclude anything that has more than 7 days between those two things. And so that's why we're asking you for the beginning of a Phenophase, try to get out there every 3 or 4 days. Intensity measures. These, I know, can be very intimidating for a lot of people, and not everybody is comfortable providing them, and that's okay if you just really can't do it. Your yes and no data is great without the intensity. However, if you are able to at least report on the low end, like if you see you're starting to see flowers, but there's only 1 or 2 of them, and you say yes to that, if you could also say, yes, I see flowers or flower buds, and there are less than 3 of them, click that less than 3 button. And I'll give you an example. For colored leaves, we have a researcher who is trying to look at the beginning of autumn and when that starts, and link that to the growing degree days and these sort of climatic factors. And when you look at colored leaf data in our database, lots of times you see the yes for colored leaves start, kind of midsummer, because you might have 1 or 2 leaves that just change color, but they're only 1 or 2 leaves, and they stay on that plant as colored leaves for a very long time. And so that makes it really hard to pinpoint when leaves like really mostly start changing color. So if you're looking at your colored leaves and the intensity bins are too complicated, that's fine. But if you know that there's that less than 5%, it's just 1 or 2 or maybe even 10 leaves on a big tree, and it's a very small number, go ahead and just click off the less than 5%. And once they start getting, the percentage starts getting higher and you're uncomfortable, that's fine. You can just stop doing the intensity. But getting those low ones would be really useful for some Phenophases in particular. Okay, now I think I'm ready for what not to fret. Okay, so I already talked about those of you who do have a scientific background, don't think too hard about what the words mean. Realize that it's geared for a more general audience. Also, don't waste valuable time poring over a single plant looking for 1 flower or 1 fruit that might be lurking, that they're really hard to see. And you don't need to catch that very first hidden flower. We're really interested in the more general patterns in the start and end of these life cycle stages. And if you miss the absolute start of the very, very first open flower, that's okay. Don't worry about it. You'll catch it next time when there's like half a dozen, and they'll jump right out at you. So don't waste time worrying about that level of detail. you just make yourself crazy. And in particular, there are a couple Phenophases in particular that you shouldn't worry over much about pinpointing the beginning. That includes the flowers or flower buds phase and the fruits phase, because in many species, both of these Phenophases start out with either very small structures, like tiny little flower buds that get bigger, and you won't necessarily catch it at the very beginning, or the signs are really subtle, like when a flower senesces and it's the ovary of that flower that turns into a fruit, and it'll start very slowly getting bigger and bigger, and it's really hard to tell once that swelling really starts. And people who are looking at the data for species that are like this, they understand that. They understand that, you know, whereas somebody that's a botanist and really understands these processes might see fruit at that very beginning, but those of us who don't understand that, we're not going to get that it's a fruit until it's like, you know, this big instead of that big. So don't worry too much about pinpointing the beginning of those two Phenophases, flowers or flower buds and fruits. Okay, I think the next slide. So there's a couple common errors. I could only think of one off the top of my head when I was preparing these notes, but the one very common error that we see is observers reporting either increasing leaf size or breaking leaf buds all throughout the year. And these two Phenophases are really only meant for new flushes of leaves. And so when we say breaking leaf buds, we mean that you know, a bud that has been sitting dormant on a tree for a period of time suddenly starts to break and you see the leaf tip. And increasing leaf size, it's, you know, a whole bunch of buds at the beginning of the spring will start to break, then you see the leaves, and then that first big flush of leaves, they just start to get bigger, and then they get to be full size and they stop growing. That's when you should stop, when you should stop saying yes to increasing leaf size. And in most cases, this, or in the north part of the United States, it usually only happens once a year. If there's an insect defoliation, then it loses a lot of leaves. They might open new buds, and that would be a new flush, and you could start using these Phenophases again. But all trees will continue to put out leaves at the end of their branches all throughout the summer, and it's not that continuing growth of a bud that's already broken and a stem that's getting longer. We don't care about that. You don't need to report on that. Okay, and tricky situations. There are a number of tricky situations, one of which we get a lot of questions about are persistent fruits. So these are fruits that they got ripe and they stay on the plant and they stay on the plant and they stay on the plant until the next season, and they're still on the plant. And, you know, they eventually start looking kind of old and maybe inedible. And where does that stop? Where does the point of ripeness become inedible? And we don't really count that anymore as a viable, you know, a fruit that some animal would want to eat. And that's really hard. And I don't have a good answer for that. Just that you're gonna, and there's a lot of gray areas like this with other, and many other Phenophases. And you are just gonna need to make a judgment call yourself. We try to cover this type of situation in the Phenophase primer, but inevitably you're going to come across something like this during your observing, and we're not going to have a concrete answer for you. We're just going to say you're going to have to do your best to figure it out. And in cases like that, we're just going to recommend that you discuss it with your fellow observers and try to at least be consistent within your group and the site that you're working at. And that's, I think, the best we can do until we do get concrete answers, maybe for some species and some situations. But for now, don't fret it too much. Just try to do your best.
And I just will add that I know that makes some of you uncomfortable not having the actual answer from us or yourself. But again, remember, it's the big picture, and what we're trying to capture here is from bigger areas, swaths of change. And so having those questions with the minutia in them isn't necessarily a showstopper. So that's why we also allow you to put unsure in the database, so that we know that you're still looking at it, and then you can move on and come back to it once we do have an answer.
So Lorianne, do you want to talk about the next few slides here?
Sure. I have to unmute again.
Lorianne stuck these slides in here. I'm like, oh, what do you want me to say?
Yeah, I just wanted to point out, because in the local phenology leader course that I teach, several people ask questions about this, either about the order of things that are listed in the protocol, because you'll see if you look at our definitions, in a linear order, it's breaking leaf buds, leaves, increasing leaf size, colored leaves, falling leaves. But essentially, some of those things, depending on your plant or whatever functional group of plants you're looking at, may happen at the same time, or they may happen out of order. So somebody asked me the other day about, I forget if it was, I think they were talking about the red maple, how the red maple gets flowers on it first before it gets breaking leaf buds on it. And they were confused because flowers wasn't the first thing that was listed on the list for them to check off. So I just wanted to point out that these things we got to list them in some way on the website, and they just happen to be in a certain order, but that doesn't necessarily mean that that's the order that those phases will occur on the plants that you're observing. And it also doesn't mean that each of those things is independent of each other. So it's not like you're going to say no to the end of breaking leaf buds and then your next answer will be yes. So you're going to be reading each of those definitions separately from the context of the whole plant. Does that make sense? How did I do, Elle?
Yeah, and I'll just add to that, yeah, these are not necessarily, we put them in an order because we have to put them in some kind of order,
Right.
for visual, or but they don't necessarily unfold that way. I mean, generally with flowers and fruits, you're going to have your flowers or flower buds first, and then you see the open flowers, and then they'll release pollen, and then they turn into fruits. I mean, that part,
Right.
I think, is pretty universal.
Right.
But certainly, whether the leaves and the flowers do not always occur on plants at the same time. So those things are going to be very independent of each other. And I'll also add that we are working on the section 2 of the Phenophase primer, where we're going to go through. We have several examples for each of the different functional groups, and we will show these very same kind of visualizations here for that particular species. And then you could more clearly see for those examples that we decide to include what typically, the order that these things typically happen in over a season.
Right. Sounds good. And just really quickly, Suzanne had a question about how best to document this. Suzanne, were you talking about this concept that we are discussing right now, or were you talking about the previous concept? So if you just type me that answer, I will answer that question in a minute. Okay.
Oh, maybe you mean what your decision was on a tricky situation. Yeah. Okay. Yeah, that's a good question. I think we would appreciate an email if you have something like that you've come across and you've noticed this, and let us know what the species is and everything, because we have, what do we have now? I forget how many plants. I think we have almost 1,000 plant species, if not more, right now. And we can't have like special rules for every single one, or we don't have the capacity to archive, to research that and archive it and everything. But if you guys have one that you figured it out, that would be great. You can send it to us, and we might decide to put it. We do have a place on the profile pages that says special considerations for monitoring, and we might be able to add it up there as, there was a group in this site, this part of the world, that saw this, and this is what they decided to do. So I hope that answers the question.
Yeah, I think so. Good.
Okay. Okay. Lorianne?
Yes. Are you on the next slide? Can you see it?
Yeah, how often and when does that?
Yes. Do you want me to answer that, or do you want to?
Well, I think I know what you mean by this, so I can answer.
Yeah. Go ahead.
Okay, so how often and when to stop? Okay, I already mentioned that we want to get the no's before you hit the first yes on the Phenophase. And so you start a bit earlier than you think your first I mean, I live in Maine, and so for me, I'll get out there, well, probably today. Actually, March 1st is usually the day that I start to go out and start looking, even though it's well before anything usually happens. But I start and I keep an eye on it, and as it looks like it's getting closer, I'll start going out every 3 or 4 days, to try to catch the beginning of breaking leaf buds or flowers and flower buds for red maple and those things that flower first. And if you live in the desert, it's going to be different. You know better than I would about, it has to do with precipitation and whatnot. And then when to stop. So for me, it's easy because I live in Maine, like I said, and once the last leaf falls off the tree, stuff is done. And so I can stop for the winter. I'm done. Once all the leaves have come off the trees, I stop reporting, and I have a break of maybe 3 months from sometime in December until March, when I just don't even bother to go out. If you live in a place where you have activity all year, and I don't watch animals either very much. If you're watching animals over the winter, then that's a different story. If you live in a part of the world where the growing season doesn't just stop for plants, it keeps going, then it'll depend on the exact plants that you're watching and when you can stop. Does that answer your question, Lorian?
Yes, I think so. And yeah, because Suzanne then also followed up with when to make a decision regarding when to stop calling it a ripe fruit, etc. And
right.
I think you said
I have a couple.
Yeah.
I have the catkins on my white birch that they hang up there, and I know they're ripe, and they're sitting up there, and they're sitting up there, and I just continue to do it until I'm done with everything else, and then I'm just like, I'm done. I mean, and I don't know of anybody that's doing a particular research question on birch catkins. If that was the case, if there was we had a campaign going, then I might pay a little bit more attention. Or if I had a reason, some of you at your local, at your local projects, you have a reason for particular species or particular phenophases, and then you can encourage people to keep going. But yeah, sometimes if it's just too much, then you can just stop.
Right. Yeah, and again, that speaks to the generality of what we're trying to accomplish here with the database, where it's designed so that people can use it locally to answer specific questions if they have them. So if you do have a researcher locally that has a question and they are interested in something more specific, then by all means, follow those rules locally. But from the big picture, if we're talking about building the big database, then I think it's less important to worry about stuff like that without having a really specific research question to answer. Okay.
Yep.
What's the goal?
Okay.
Yeah.
I can read this one again.
Yeah.
We're hammering this one home for you guys.
Yeah.
What's the goal? Remember, protocols are to be taken at face value and designed to be reported consistently. The goal is to give the people that use the data, a valuable picture of what's happening on the ground with the plant or animal species throughout the season, and not the details of on any one day whether or not there was 5 old leaves and 20 young leaves. Don't worry about that level. Just go through quick. It's better to have, we would rather have more data. We'd rather have data on more species than more data on one plant. Yeah, I covered that one.
Okay. And I see a question here from Russ. He says, there are 9 people in my group at my plot. I try to drive the hour and a half every other week. Is that enough? Even though I have never seen anyone else in my group at the plot. So I'll answer that question. And I would say, if you have a group of people that are sharing the workload of doing observations and you can affix it so that there's somebody out there at least weekly, I think that is a good goal. So you personally don't have to be the one to go out there every week to observe. And then again, depending where you are in the country and what plants you're observing or animals you're observing, you're going to want to make sure that you're capturing the transitions between the phenophases. So you might want to set your schedule so that there are people going out twice a week instead of just once a week, depending on what you're observing. Okay, let's see. So here we are on the questions now. These are some of the specific questions that y'all have emailed us, or some that we had kind of in our back pocket. So I will read the question and then Ellen will give us the answer. First one on our list is, your site-based protocol says that we should observe 3 plants. What if I don't have 3 plants of that species at my site? Does that mean I can't observe?
Not at all. Information from a single plant is just as valuable. The reason that if people have the time, and if they have that many plants at their site, you can see variation between individuals in the same species. And so I, for instance, have 3 forsythia that I monitor, just because I happen to have 3 different forsythia plants in my yard, and they're generally in sync. And so you know, I get most of the information I can get if I was only doing 1 plant. But every now and then, in the fall especially, or the late summer, a couple of them will throw a couple extra flowers, but the other one won't. And so it's sort of, you know, if you have more than one of the individual at your site and you have the time to monitor it, it's sort of interesting to see that variation. And I noticed that we have a bunch of people from Neon here at Neon sites, and that's what these guys do. They're looking for that variation across the species. And so I think in cases they have like 30 different individuals that they're monitoring of a single species, but they're paid. Those guys are paid to do it. And for those of you that are just volunteers, yeah, do not fret that you only have 1 plant that you're watching, because we get, you know, I'd say 75% of the information we get from just the 1 plant.
Okay. Next question. I'm new to this. This is my first season observing, and I've noticed that it is taking me a long time to answer all of your Phenophase questions. Some of them I have trouble identifying, especially the intensity. Can I leave some of the observation fields blank, or is my observation incomplete if I do not answer all of the questions?
Especially when you're first starting out, do not worry about answering them all. I mean, we even tell people that, you know, you don't have to answer any of them. If you just sort of go out and look and start to get a feel for it without reporting anything, that's fine if you just don't feel comfortable reporting. And absolutely, there are some, for instance, forsythia again, it's really hard to see the fruits on forsythia. And so I've given up, because I don't think anybody cares about forsythia fruits. I've given up. And so every time I go out, I do not circle anything for any of the phenophases for forsythia. And that's perfectly reasonable to do that. And intensity too, if it's just too confusing, don't do it. But like I said, on the low end, if you do see a flower and you know there's only 1 of them, then go ahead and click that less than 3.
Yeah. And what I usually tell people, especially if they're working with groups of observers, is to take the least common denominator of the definition. So if the most easy things to identify, if you're talking about a deciduous plant or a flower or something, do the leaves, the flowers, and the fruits. Don't worry about breaking leaf buds. Don't worry about any of those other slightly more complicated things until the group of people is comfortable identifying it. They understand what that looks like on that plant. They're actually observing the right plant. And then that might even take a whole year for you to get to that point where you're comfortable with knowing what the difference is between certain things on the plant and certain phases, and that's perfectly fine. So that's why Nature's Notebook is a little bit more of a long-term commitment than some of the other citizen science programs out there, just by design, because of the way that we're trying to capture the transitions between the phenophases and have data in specific locations for more than 1 year at least.
And I'll also add to that, feel free to, it's better to have no data from the standpoint of the data quality stuff that I talked about earlier. We're better off having no data for a phenophase than having wrong data. So don't click the no if you're confused about it. Just leave it blank, or click the question mark. But don't fill out no's if you're not really sure. Just feel free to leave it blank and not check anything.
All right, we have had difficulty observing initial growth on a perennial forb we monitor. The definition states that growth is considered initial on each bud or shoot until the first leaf has fully unfolded. But no matter how young a SOAL6 seems, there is at least one leaf fully unfolded on its single shoot.
Yeah, this is a great question. Some species, in some years, some species all the time, some species just in some years, they develop so quickly in the spring when it starts to warm up and they start putting out their leaves that you might never see the initial growth stage. You might just there was nothing, and then you go out 3 days later and it's already has leaves on it, and that's fine. If you don't see it, you don't have to ever report yes for initial growth. It could happen in 2 hours, and if you don't happen to be there in those 2 hours, you're going to miss it, and there's nothing to be done about that. So that's totally fine if you completely miss a phenophase. And like I said, some regularly do this. In fact, I think for some of actually it was the breaking leaf bud thing for the species in the desert, they just go so quick from no leaf, nothing, to suddenly a big leaf. It goes so fast that we eliminated that sort of early stage leafing phenophase from all the protocols.
Okay. Let's see, the next one is, it takes me a long time to count all of the flowers at the top of the tree, which I cannot see very well. Is there a better way to get that information?
Yeah, so if you're working with tall trees, especially you're in a forest and there's no lower branches. The only thing that you have available to you is way up high. I use binoculars, but even then for tiny, tiny structures, especially some flowers in oak trees, the female flowers in oak trees would be impossible. And so you can ignore the counting if it's just too difficult because they're too small. But if you do have a tree that does have lower branches and you can see, we're not asking you to count all the structures on the tree in those intensity bins. We're just trying to get a ballpark estimate. And the way you can do that is you take one branch that you can see, and you kind of eyeball how many there are. One small part of a branch, one twig, and how many flowers are on that. And then try to extrapolate how many of those twigs are on the tree. And let's say you had 10 flowers on your little twig, and you're going to estimate that there are 100 twigs like that. So 10 times 100 is 1,000.
Yes.
But there's probably more, and you'll easily get over 10,000. I find that all the time in the big trees I do. You quickly get over 10,000 when something's in the heat of starting up in the springtime. And so, yeah, really just don't worry about counting the whole thing. Take one little part and kind of extrapolate, and it takes practice. It's going to take practice to do that. But that's the way we do it.
Yeah. And the other important thing I think to note is to make sure that you're doing it consistently. So if that's how you've been counting it the first couple times you went out, make sure that you're counting it the same way, regardless if you're feeling like you're 100% correct or not. So just make sure you're doing it using the same process.
Yeah, and this is another thing that's great for, especially if you have a group of observers that are doing the same plants. Make sure you guys are on the same page, because really the value of knowing the counts of flowers and fruits, it's really to see if there's a change from year to year on that tree at your site. And so this thing, I think with this particular, the counting of the flowers and the counting of the fruits, we're not really worried so much whether your red maple in Alabama is the same as my red maple up in Maine. We really just want to see from year to year, do you have a low number of fruits, and then suddenly you have a large number of fruits in 1 year. So that's why these estimates it's okay just to do the estimate, and as long as you're consistent at your own site or on that individual tree, then that's good enough.
Good. Okay. Let's see, the next one is, my plant's natural history indicates that the flowers appear before the leaves on the plant, but your protocol says that breaking leaf buds is the first thing to report on the list. Can I report my answers out of order? We touched on this a little bit, but if you want to.
Yeah, we pretty much already talked about this. Each of those phenophases is meant to be evaluated individually. There are a couple that are nested. So if you see open flowers, then you are also going to be seeing flowers or flower buds. But especially between the leaves and the flowers, they're completely independent of each other, and it doesn't matter what order they come out. Just report it as you see it. If you see leaves in a certain phase, then say yes. If you see flowers in a certain phase, say yes. And they could very well be on the tree at the same time.
Okay. So these are kind of two similar questions, so that's why I put them in the same slide. The one on the left is, in the springtime, the first thing I see on my tree are reddish leaves. Do I report these as colored leaves? And then the one on the right side of the slide is, the maple leaves turned yellow with dark spots in late August, and the dawn redwood needles turned brown. There's a locust tree which also has similar symptoms. Other trees in my neighborhood do not show these signs. I had hesitated to enter the color change for this reason.
Okay, yeah, so this is a colored leaves thing, and reviewing the definition for colored leaves. One or more leaves show some of their typical late season color, or yellow or brown due to drought or other stresses. And I know that for red maple, they do come out with this reddish color, and that hue is also typical of the colored, or of the fall senescence, but that's not what we mean. When we're talking about colored leaves, we're really talking about leaf senescence, and when it starts to, it's basically losing its green color, losing chlorophyll, and showing the other pigments that are there that you don't normally see during the major part of the growing season, because it's hidden by the green. And so, no, do not report colored leaves for those initial things, just new leaves coming out. And then the second part of the question, let's see. Do not include spots. Okay, so this says don't include spots. I think I had a different answer. Oh no, here we go. Okay, so this one, yeah, so this was somebody, so the second part of this question, the slide, was somebody that had, it sounds like their tree was declining, getting sick, and other trees in the vicinity were not, and they were worried about reporting what they were seeing on this tree because it was atypical of what was happening. And I think, you know, the thing is, we're asking you to follow an individual, and if an individual gets sick, then yeah, you just have to report what that individual is doing. And so this case, I would say, yes, you can report the colored leaves on that. But it helps if you put in the comments that you think this tree is sick. And that's why you're seeing this sort of colored leaf situation. And if the tree continues to decline, you know, after a year or two, then you might want to drop that tree and choose a different individual to monitor. Because again, we sort of want to know what typical healthy trees are doing, not an individual that happens to be sick. If all the trees are sick, then that's actually really important to continue to report that, because then we know something's going on. You know, that people want to know, and management, you know, do some sort of figure out why is it sick, and help the trees in that part of the world get rid of their pathogen or whatever is going wrong.
My deciduous tree keeps putting out new leaves all year at the end of each stem. I've been reporting breaking leaf buds or young leaves for this. Is this correct?
Yeah, so we addressed this earlier. No, that is not correct. All trees and shrubs, deciduous trees and shrubs do that. They continue, the bud breaks in the spring, and then the stem continues to elongate until sometime in late summer when it stops growing. And while it does that, new leaves come out at the end of that stem the whole season. That's not what we're trying to capture with the breaking leaf bud and the increasing leaf size phenophases. We only want to know of that first flush of leaves as the canopy fills up and becomes what it looks like full when it's in middle of summer, once it hits that point, that's when you stop saying yes to increasing leaf size.
Okay. Let's see, this one is what to report on plants that have phases such as buds or fruits that stay in the bud phase for a really long time.
Yeah, we talked about, we didn't talk about this. We talked about persistent fruits. Yeah, so we don't have, there are some cases, but if buds are dormant, if they stay in a bud phase because they're dormant or resting, then we're not observing those at all. We're just letting them, we're not reporting any phenophase for those. If they're flower buds that are kind of just resting and they're waiting for, there's no change, that you don't see any change as you go back week after week, don't report anything for those. They're eventually gonna break or start swelling, and that's when you can start reporting activity for either, if they're leaf buds, you'll see the breaking leaf bud when you see the tip. If they're flower buds, once you see them starting to get bigger and looking like they're gonna burst, you'll start saying yes to flowers or flower buds. And then with the fruits, yeah, there are some species that they've got fruits from several years still on the plant, and we have no way, we have not, nobody's decided this was important enough to for us to come up with separate phenophases for first year fruits versus second year fruits versus third year fruits or whatever. So if you see a ripe fruit or on the plant and you've got both new ones and old ones from different seasons, just say yes and count them. In the note, if you want to, in the comments, mention that, you know, half of them are from the season before and half of them are new, or whatever the ratio is. That would be fine.
Okay. Suzanne has a follow-up question about the breaking leaf buds from the previous slide. She says, but I thought the protocol says to report breaking leaf buds throughout the growing season. And I can't open the protocol because it'll mess up the slides. So I can't read it.
Yeah, so, okay. So for breaking leaf buds, let me see if I happen to have it right here. Oh, I might. Okay, breaking leaf buds, I'm gonna read what I've got here. One or more breaking leaf buds are visible on the plants. A leaf bud is considered breaking once a green leaf tip is visible at the end of the bud, but before the first leaf from the bud has unfolded to expose the leaf stalk or leaf base. Maybe she's talking about somewhere else. So, okay, so leaf buds, it's possible for leaf buds to continue breaking throughout a season, or to break not just at the beginning, but later on in the season. But what you're looking at for that is you're looking at the bud that's got bud scales, and this is a naked bud. You're not looking at the thing at the end of a growing and already partially growing or elongating twig. That's not a bud at the end. That's just continuing to put it's just like growing tissue that continues to put leaves on. What we're talking about is that little thing that's, I mean, they come in all different shapes, but you know, kind of like this shape, sits on the stem, and often they have leaf scales around them. And once those things break open and you suddenly see little green beginning of a leaf coming out, that's what we're talking about for breaking leaf buds. And it can happen throughout the season, but just make sure that you're looking at a bud that's breaking and not the end of a twig that's just in the process of elongation.
Okay. All right. Let's see. Maybe this is the same thing. Can I see leaf buds on my tree in the fall or winter? These are breaking leaf buds, right?
I see leaf buds on my tree. These are breaking. No, probably not. Okay, so yeah, so leaf buds will sit there on the stem. They're going to be dormant for a long time. The way this happens in a lot of tree species is they'll set buds in the late summer. So that stem that's been elongating and putting out leaves, it's going to stop growing in the late summer, and it will start to form a bud at the end. But this is not what we're talking about. That's the beginning of a bud. And it eventually, at first it looks sort of like green and tender, and then it'll look a little harder. Either it'll have bud scales on it and be like a brown color, or if it's a naked bud, I've never actually watched those form. I'm not sure how they look in that period in late summer when they're forming. But that, we're not talking about that. That's not a breaking leaf bud. A breaking leaf bud is one that has been dormant and then is in that process of newly opening and starting new growth, which usually happens in the spring. But like I said, if you have leaf foliation or something like that, you can get these buds that come up on the branches that are starting again. Let's see, I had some other notes in here. Essentially harden. Yeah, yep. Or you could have, yeah, sometimes if you get a really warm fall, you'll have the winter bud will have set at late summer, but you've got really warm temperatures in October. Occasionally they will break and you'll get another little flush of leaves, and you can report as breaking leaf buds.
Okay. Let's see. How do we explain or take into account external factors that can affect data that have nothing to do with climate change?
Okay, was this related to Bettina's question about,
Yes.
okay, yeah, so Bettina wrote to us about, I forget exactly, but something, I think they cut trees or something near the bird habitat that they've been watching, and it's affecting the timing of various things going on with the birds. And really the only way we have to report stuff like this is in the site comments. And so I would recommend that you go in, if you suddenly had a clear cut next to something and that's going to change everything, go into the site comments and put the date that it happened, and just a relatively brief description of what it is, because I'm not sure how many characters that field holds of what happened that might be affecting the phenology at your site. That's the only way we have to document that sort of issue.
Yeah, sounds good. Okay. All right, now we have a couple more specific questions, and we are almost at the top of the hour, so folks have to go. Like I said, we are recording this. I think we only have maybe 5 or 6 more slides left, and then Ellen and I can hang out for a little bit longer, not more than an extra half an hour probably, but if you have questions, I'll open up the microphone for people to ask them when we get to that point. So here we go. Is the intensity based on each flower or on the inflorescence as a whole, which means single flower versus catkins / inflorescence?
Okay, yeah, so this is somewhat outlined in the definitions for these things. I'm going to just briefly, let's see, flowers or flower buds. How many, yeah, okay, so it's in the intensity question. How many flowers or flower buds are present for species in which individual flowers are clustered in flower heads, spikes, or catkins? Simply estimate the number of flower heads, spikes, or catkins, and not the number of individual flowers. So that's going to depend on the species, and unfortunately, we just don't have the resources to do that kind of research for the 1,000 plant species we have and write specifically for each one what you're counting. And so you unfortunately are going to have to do a little bit of research on the plant that you're looking at to figure out whether it's considered an inflorescence or not. And yeah, there are some that are just, plants are so complicated. They're so complicated. And some things it's easier to say whether it's an individual flower or an inflorescence. Others, like maybe it's in botanical, technical terms, it's an inflorescence, but to you and me, who's not a botanist, it looks like an individual flower. And we have not cracked that yet, not yet. I'm not sure what to recommend to do there. I think the best thing you can do is just make sure at your site with your group that you're consistent. And you could certainly email us and let us know that you decided this for this particular species. And we could then perhaps put that in the special considerations for monitoring for that particular species. So that's the answer to that one. For open flowers, it is always the individuals that you're counting for the intensity, regardless of whether it's in an inflorescence or they're single flowers. You're always looking at the percentages for the individual flower, and it actually makes it really easy if a flower is in an inflorescence, because what you can do is you just take one inflorescence and you count the number of total flowers, and then you count the number that are opened, and you divide the number of open flowers by the number of total flowers on that inflorescence. And then as long as it looks pretty much the same across the whole tree, you've got your number right there just by counting the flowers on one inflorescence.
Yeah, okay.
I think I read everything here. Yeah, got that.
Nice. Okay, the next one is about grasses, everyone's favorite difficult thing to observe. I am confused about little blue stem flowers, and I can't find the information I need on the web. I see fluffy tufts, in parentheses, spikelets. Are these flowers? I also see anthers and fuzzy stigma. Do I wait to see these before saying I see a flower or a mature flower?
All right, I'm going to start off by just saying grasses are really hard, and I personally have not spent a lot of time observing grasses phenology, and I'm not familiar with little blue stem in particular, but I did do a little research for this question. We do have some detailed information about grass flowering in the Phenophase primer, so you can look there. Your species might not be representative, but it gives you sort of a general idea of what you're looking at. And I did look up blue stem, and I think those fluffy tufts that you described, they're hairs that are part of the spikelet, and they become more obvious as the season progresses. They're part of that reproductive structure, but looking closely at those hairs is not going to tell you anything about whether the flowers are opened or about the state of the grains, the fruits that are ripening. And so what you really need to do for the Phenophases, you can start saying yes to flower heads. Grasses have a flower heads Phenophase and open flowers and a pollen release Phenophase. For flower heads, you start saying yes once you see that spike. It looks different than grass leaves, comes up out of the middle of the stem. Then you got a flower head, and you can keep saying yes to flower head. And then once you see those anthers, and whoever wrote this question says they see them, so they know what they're looking for there. Once you see the anthers or the fuzzy stigma, then you're going to be answering yes to open flowers, because you are seeing those particular reproductive parts. Once those wither, the reproductive parts wither, then the flower is senesced and you're no longer seeing open flowers. And you can also, once on the whole plant, once all the flowers are done on the whole plant, you can stop saying yes to flower heads, because at that point, we're switching from flowers to fruits. And then I have not done this myself, but if you're good at it, I'm sure it takes practice, you can kind of figure out whether or not those flowers had been fertilized and the grain is developing, and then you can say yes to the fruits. And then once they get hard, I forget what the definition says in here, I don't have it up. Once those get harder, then you're going to be seeing ripe fruits.
Yeah. And again, if you're not sure, you can just skip that until you do find the information.
Right.
Fret not. Okay, the next one is, I'm having trouble distinguishing between buds and seeds on Forsythia. Understanding the timing and appearance and disappearance would also be helpful.
Yeah, so I told you about my Forsythias before. So on the timing on this, you're going to have the dormant flower and leaf buds are going to be present on the plant over the winter. And you can start saying yes once yes to flowers and flower buds. Forsythia is one of those things that flowers first. And so you'll start to see these buds swell. If you've been watching, you see nothing, nothing, nothing. You'll start to see them getting a little bit bigger, and you'll start to see a little bit of green as the bud scales slide apart. You can start saying yes to flowers and flower buds. And then some of them do have only leaves. Those usually break later. You'll see the green leaf tip. Once the flower buds start to slide apart and you're starting to see the flower petals, that'll be yellow in color. And then once the leaves go, those will be bright green, just like regular breaking leaf buds on other trees. The fruits are going to develop after flowering, sometime in the summer. And I think like I said, they're really hard to find because they're the same color. Like as mentioned, is the stems and the buds, and they're kind of lost in the leaves there. I think that they do break open though, and they drop off, or at least they drop their seeds, and they're no longer count as fruits sometime in the fall, summer to fall. So that's the timing of it. And I feel like we have a picture somewhere, but maybe
yeah, we may have a picture in the primer, the Phenophase primer.
Yeah, we might. But anyway, like I said, I stopped bothering with fruits on forsythia because they were too hard to see. I was spending too much time trying to see if there was like 1 or 2 stupid fruits on this plant, and it just wasn't worth it. So you might want to consider that.
Yeah. And I think for this description that you just gave, Elle, and the question that the person submitted before about the doing research on the plant, I think it's also really important, an important part of your observation practice to observe your plant, but also to study up on the natural history of it to kind of get a sense of generally when each of the phases will happen, what do each of the phases look like. You might want to do that before you even go out and start observing so that you know what you're looking for, instead of just jumping right in to read the Phenophase definitions and expecting that to answer your question about that particular species.
Yeah, and I'm sure Lorianne has told you this before, and I'll just add it here. If you do a search, you put in your species, scientific name, life cycle, you often will, Google will give you some great information. Go straight to the extension websites, because they often have really great information about that. You know, there's a couple that I've run into. There's one in Minnesota, I think. There's one in Illinois and Massachusetts, and so there's a bunch of them. Depending on the species, you might get really good information about that.
Yeah, and the extension offices in your counties are usually ready and willing to answer questions. If they have enough staff and a horticulture agent on staff, they can help you answer the questions about plants or plant identification. Because again, we don't have the resources to help everybody identify all the plants in their landscape and understand, you know, what the differences are between, you know, different varieties and that sort of thing. We're kind of looking at the big picture, the general picture. So an extension office can be a really great resource. Okay. The next question is, one of the species we will be observing is the honeybee. We have several apiaries on the property. Do we count the activity in there when we are making observations? I was most curious about larvae and eggs. Is it okay to report on honeybees that were part of an apiary with our program?
Yes, absolutely. You can report on honeybees. And in fact, the protocol that we have was developed specifically for for beekeepers, because we do include stages for eggs, larvae, and drone cells, which you wouldn't see them unless you have a beehive and you're pulling out the drawers and you can see the combs. So absolutely, you can report on that activity. And I think that this question might have, they might have thought that this was a sort of artificial habitat, which is true. I don't know much about beekeeping, but if you heat them, if you add heat, then you might want to just add that in the site comments, or in the site comments, because that will change the phenology relative to just the ambient temperature in the outdoors. So if there's things like that you do that are managing it separate from just being outside, include those in the site comments.
Mm-hmm. In the same way that the site comments also take information in about whether or not plants that we have are fertilized or watered or something like that, just so we have a general sense of what's happening at that particular site.
Yeah. Mm-hmm. Yeah, the one thing we have for plants, you can, there's a couple fields, and there's just a free text box for individual plants. You can put in a plants comment. For the animal species, we don't have that. You can't, we can't submit comments at the animal species level. So that's why I say for this case, if you got bees, put it in the site comments.
Yep. Perfect. And there's a note on the chat box from Laura regarding the previous question about the forsythia, which I think fits in with kind of what I was saying, but she says that a lot of the forsythia cultivars in the US are sterile, so they may not ever produce fruit. And that might be one of the reasons the person had a question, if they couldn't ever see the fruit. So I think that probably is true for a lot of the landscape plants that are sterile, because especially things that are relatively invasive, like fountain grass or something like that, and the nurseries are selling sterile plants, you might want to think about that too. Not that it matters necessarily one way or the other for participating in Nature's Notebook, but it might just not be producing fruit or seed if it's bred to be sterile. So good question and comments.
Yeah, good point. I did figure that was why mine had very few fruits on it. I think things that are sold for because they have showy flowers, especially, or leaves, they may not be sterile.
Exactly, right.
Or they might be sterile, yeah.
Okay, so we can just take a couple minutes to see if anybody has any questions that they want to ask. And I think I can unmute everyone. I hesitated to do that because at our top participation, we had 60 people on the webinar, and I knew if I had that totally open, somebody would inadvertently have left their microphone on. But you all can feel free to either type something in the chat box now if you like, or raise your hand. I think I should be able to see that, or let me know that you want to say something aloud and I can unmute you specifically if you have a question. Okay. I see Georgia asking a question. She says, can you report on an animal if you watch them via a live webcam? That's a good question.
Yeah, that is a good question. And we have, for the animals, we do have Phenophases for capture method. So for instance, for butterflies, we have individual at a feeding station, individual in a net. For some of them, we have individual in a trap. And those things with the animal, for animals, a big thing is about the detection probabilities. Unlike plants, which are there, and if you look closely, you'll see it. The Phenophase is on a plant. With the animals, you may not see the animal because it's not there. It might be hidden, and you don't know it's not there. And so that's why we had those methods in there about detection probability. So catching it on a webcam is definitely one of those things that you might not have seen it, but because you have your webcam going, you'll see it. So I think, yes, it's okay to report that. But we are thinking of adding that. If people want to be doing that a lot, we could add that method, I think. So I'm going to say yes, you could do it. Let us know that that's what you're doing, and we'll decide if there's a need to do that. And add it as a okay. So we're about to get a site visit comment box. And I think that would be the best place to add that once we get the site visit comment box. So on that visit, for that observation of the animal, you can say that this was captured with a webcam.
Can I add more context?
Yes, that's why I unmuted you, Georgia.
Yes, yeah, great.
Okay. I think I know what you're going to say, but go ahead.
So it's a nesting pair of red-tailed hawks. They have been nesting on a window ledge of the university library in my neighborhood since 2011. It's the same male. This year it's the third female. And so they've been repairing the nest already. And it seems like, people are sort of waiting for the female to sit on the nest overnight. And that's usually the sign that she's about to lay eggs. So I haven't been keeping track of them. But it just occurred to me that because I've been focused on plants, but I thought I could use our Nature's Notebook account to track the hawks. And I was just curious if it was okay to use this webcam versus being in the park with my binoculars capturing the data. And then I guess how would it, I've never used Nature's Notebook. And so for an animal like a hawk with a webcam on its nest, are you doing then a daily monitoring? How does the frequency look for a situation like this?
Well, yeah, I mean, we haven't added in this type of specific monitoring to Nature's Notebook. There might be other places that do that kind of thing. I'm not sure, but my recommendation, if you wanted to add it in Nature's Notebook, is yeah, I mean, you would have to enter that data yourself. And so if you want to go through the webcam every day and put an observation as to whether you see this, that, or the other thing, I think that's fine. And just you put it in on the day, and maybe even the time. I mean, you'll have a timestamp too, so you could add the time as well. Yeah, I feel like that's up to you, and how much work you want to put into documenting, like sort of basically transcribing data from this video feed to boxes in Nature's Notebook.
Yeah. And I think the only other thing to think about, and maybe you already thought about this, is you probably need to create a new site in Nature's Notebook, because if that's not where you're doing your observing for your plants, you would want to create a site for where the camera is. That's what I was originally thinking. If somebody was going to try to record some data from one of those Cornell webcams or something, they'd need to know what the location is because it's attached to the site. So that would be the only.
Oh yeah, perfect. Right, and then that could go in the site comments. That this is I would even name the site that, so that people would, that would be right off the bat. It's such and such webcam.
Right.
Yeah.
Yep. Good. Okay, Bettina has a question now. Let me unmute you, Bettina. Hang on one second. I can find it. There we go. Okay.
Good morning. Thank you. You know, it really goes back to my original question, and it's a simple one. You keep mentioning the site comments, but is that something that's only opened up to administrators? And is that something that is under the Sea and Sage? Like the site.
I'm going to check here. I thought there was a site comments box.
No, on the animal protocols, there's only a little notes that you can put for each individual species, and so we've been putting notes, you know, whatever kind of notes that we might have observed about each of the species during our survey, but we don't really have a full box that we could write. You know, we had XYZ raptors occurring during our survey, which clearly would affect their behavior. We had, you know, predation at the bridge where our cliff swallows are, or in the case of my original question, they've come in repeatedly over the last 2 years and removed the vegetation right in front of the colony prior to their arrival. I mean, days within the cliff swallow's arrival. So there's no place to really put that except in the comments for the cliff swallow. So but there's been a huge decrease in nesting. We, like 2 years ago, we had over 200 nests. The previous year we had 75, and last year we had about 15.
Yeah, I will say, Bettina, your question has actually driven our goal to add that sort of comments field on the mobile app. So that is coming very soon.
Okay.
We are told by our IT team that it should be within a month or two that the mobile app will be released. And that was the main reason we asked them for that particular site level comments. But Elle, I think we don't have it on the observation deck, do we? If you're using the computer, did you find that?
Well, so so I'm confused. I went into my personal site. I'm not part of a group site, so I'm not quite sure how that might look different. But on a personal site, if you go in to edit your site, there is a comments box at the bottom where you can add comments about the site. Right.
But Ellen, I think that's on an administrative level. I think for our participants, I don't think that that's there, because I am not an administrator and I've never seen that. But I could be added. If you would add me as administrator, that would be fantastic.
Well, we can look into that. We also, at the individual level, I know that you can put a comment in about your individual plant. I don't see anything for animals, I don't think. If you're an individual observer and you want to put in comments about your registered animals,
well, we put in a comment under each, when we put in our data for the day, we can put a note at the bottom for each species. But then if we have something that affects the whole across the board, like they're doing construction or something, we would have to make that note in every single species.
Yeah, that doesn't make sense. So we'll look into that, because you should be able to add a comment for the site.
Okay.
Yep.
And that would be pertaining to that particular date of observation, right?
Right.
Well, in
the
two different things.
Scenario, yeah.
there's two different things. There's a site comment, which in your case, I would recommend putting this at the highest level because it's something that's somewhat permanent, for at least for the clearing the vegetation. And you can put a note in there, the date that it happened. Let me finish. And then we do not have this yet anywhere, but we are planning to have a site visit comment so that for the day that you go out, if there's something like the weather or whatever you want to make note of, you will be able to do that, but that is not yet available.
Okay. That's, I guess, what we're really looking for in this situation.
Yeah.
Yeah, and that's where I was saying you guys are the drivers for us really adding that to the mobile app. So
that's fantastic. I don't know how many people I can train to use the mobile app, but that
yeah,
I'm
yeah, sounds good. Okay, thanks. So we have a question from Maria. She says, how should we account for errors in measurement estimates from one year to the next, or even one observation period to the next?
I think I'll need some clarification from Maria on what she means by errors in measurement estimates.
Yeah, let me unmute her if she's still here. Hang on a second. There we go. Maria, can you talk?
Hi, yes, this is Maria. I just meant differences in, for example, like 1 week there's bud break and the next there's not.
Yeah, are you talking about just reporting that in Nature's Notebook? Or do you happen to be from NEON?
Yes.
Oh, okay, you guys might have other levels of protocols that we aren't a part of here. Maybe is Katie Jones still on the call? She might be able to answer that.
Hang on 1 second. Yes, unmuting Katie.
Oh, well, I can always talk to her outside of this session. That's okay.
Okay.
Okay, thank you.
How convenient. Perfect. Good.
Sorry, I think I was muted on my end and not on yours, but yeah, we can talk about it offline since I think it is specific to NEON procedures rather than Nature's Notebook.
Okay.
But let's definitely touch base later.
Excellent. Thanks, Katie. Okay. And we have a question about the mobile app and what's the name of it. I was going to type it in the box, but I will just say it out loud. The mobile app is called Nature's Notebook. So if you have an Android or an iPhone, you should be able to search in either the Apple Store or the Google Play Store under the name Nature's Notebook. I think if you leave out the apostrophe on Nature's Notebook, it will come up in the search and then you can download it there. And as I said, we are getting ready to release a new version pretty soon. I don't have a specific release date on that, but once you download the app in the store, you should be able to get the update whenever it's ready. So I think that sort of thing happens automatically. Okay. Anybody have any other questions? All right. Well, on this last slide, I did want to point out that we do have our two primers. So we have the botany primer and the Phenophase primer. So those should be the first place that you should go if you have a question specific to a botanical issue or something you're not sure about related to Nature's Notebook. And then the Phenophase primer, we are still working on 2 of the other sections, but right now, Section 1 describes pretty much in detail about the phenophases, and they are done at the highest level, so we don't have, as Ellen was describing earlier, pictures of every single phase for all 1,000 species in there, but we have representative photos and descriptions for what each plant looks like. On the Nature's Notebook website, in the upper right-hand corner, there's a link to a help page. So if you click on that, we have animations on how to do things in the website or on the mobile app. And on the sidebar there, I don't know if you can see this in the small screenshot, but we also have what we call Nature's Notebook Nuggets. And a lot of these questions that we've answered are found detailed in there. And Erin, our outreach coordinator who sends out our newsletters, will pretty much every season link or 1 or 2 of those nuggets in the newsletter to the observers. So that's another place to find that. There's a link on the Observe tab on the website that is called Learn How to Observe. And again, all these things are congregated there on the sidebar, so you can get access to all the information you need from that particular point. At the bottom of every page in Nature's Notebook, there's a link to frequently asked questions. So all of the stuff that we've ever been asked is also cataloged there, including the questions that we've answered today. And then the last place you can look is the Nature's Notebook or USA-NPN YouTube channel, which is the top picture there. If you just Google USA-NPN, it should come up. So on there, you'll find recordings of all of our other webinars, information for local phenology leaders, and we have a lot of the videos and the how-to videos are embedded in there as well, so you can just watch those at your leisure. And this recording I will also be putting on the YouTube channel. And as I said in the beginning, we will forward the link to everyone in the list just as we announced that this webinar was happening. So there is no lack of information. You should be able to find the answers that you need somewhere. And if you still can't find the answers that you need in any of those many resources, you can email us at phenophases@usanpn.org. So that's your place where you can ask all of the questions. We try to answer those questions as quickly as we can. We have a very small staff, so if your question is not answered within 6, 8, or 12 hours, be patient. We're not monitoring it 100% of the time, 24/7, and it is answered by usually Ellen or myself, so be gentle with us. And if
we don't work on weekends.
We don't work on weekends. So if you email us something late on a Friday afternoon, don't expect to have it in your inbox Monday morning first thing. And the same thing, if you have questions about using the technical side of Nature's Notebook, like the web app or the mobile app, then you can email support@usanpn.org. So thank you all so much for spending time with us this morning. This was a great conversation. I hope we were able to answer some of your most burning Phenophase questions and questions about Nature's Notebook. If you have anything else you want to share, definitely email it to us. And otherwise, I think I will say thanks for participating.
Yeah, and I'd just like to add on to that, I am interested to know if this was helpful or not really helpful. So if you still didn't have your question answered, please go ahead and email that and let us know how we might do this better if we do it again.
Yep, exactly. Or if you have a fresh idea about how to communicate the information to people, by all means, we're all ears and responsive. So, all right. Thank you all so much.